Okay, I think we'll go ahead and get started. Thank you all very much for being here today. We were so disappointed when we had to cancel this event in April, but we're very excited to have it going today. In this panel, we hope to delve more deeply into the possibilities granted by CRISPR, along with the ethical implications of the technology. The format today will be to briefly introduce each panelist and have them share their main takeaways from the film. After that, we'll open up the panel for questions from the audience. We ask that you use the Poll Everywhere link that is in the chat window to ask your questions and or to upvote other questions that you would like to hear the panel discuss. We're going to use this technology to try to answer first the questions with the most audience interest. And with that, I will introduce our moderator for the day. Gail Henderson is a social scientist and professor of social medicine here at UNC. She was department chair from 2009 to 2015. And since 2007, she has been the director of the UNC Center for Genomics and Society, which began as a center of excellence for ELSI research funded by the NHGRI. Gail, take it away.
Thanks, Christy. So I'm delighted to moderate this panel, and it's based in a movie called Human Nature, the story of CRISPR, the most important scientific discovery of the 21st century, which is a pretty stunning claim when we're just at the beginning of that century. It's going to be a wonderful hour because we've assembled a panel of interdisciplinary scholars that are really among the very best from UNC to take on questions about editing along ethical boundaries. What does that really mean? So my job is to introduce very briefly, ask them to reflect on what they considered the biggest takeaway message, move through that. That'll be perhaps 20 minutes, and then turn the discussion over to a number of extremely interesting questions that have come in for them to take on. So without any delay, Dr. Stan Ahalt is our first speaker. He's the director of RENCI, which is the Renaissance Computing Institute at UNC Chapel Hill, but it is an institute that links UNC, Duke, and NC State and partners across the country. As the director, Stan leads a team of research scientists, software network engineers, data science specialists, and visualization experts who work closely with faculty research teams in all these institutions. And he claims that his area is cyber infrastructure and data science. And so Stan, from your perspective, can you tell us what your biggest takeaway messages were from this movie? And Stan.
Yep, sorry, I was pushing on the space bar to unmute and that was not successful, so lesson learned.
Okay.
So thank you for the kind introduction. As the least knowledgeable person on this panel about actual genetics, and I have to be very upfront about that. The biggest takeaways I had were actually two things that are linked together, at least in my own brain. One is that I was very impressed with the realization that, of course, we all know, but I think that it drove home even more fundamentally, that the basic science is so important to these major advances. The fact that pieces of the CRISPR story and the Cas9 story and how the guide sequences work had been put together by a collection of people adding incrementally, in some sense, to each other's knowledge, and suddenly, you know, there's a breakthrough in the technology, and we are capable of doing something that immediately spawns these companies, you know, that have various new economic opportunities. And to me, it was just a compelling story of the basic science transferring to a more team science setting very rapidly. But I was also struck, and I actually watched the show twice, and I think I'm going to watch it a third time, because every time I've watched it, I've gained more insights. And the other thing that strikes me, and I'll leave this till later to elaborate on so others have an opportunity, is how many questions that have arisen in my mind as a consequence of watching this movie. There's some fundamental science questions that I think I should know, but I don't know enough genetics. And then there's questions that I have arisen since I've been pondering it, knowing I'm going to be on the panel. Of course, that stimulates some thinking, that I think maybe that have not been answered yet, that I think that there's some multidisciplinary questions that need to be posed in light of this new technology. With that, I'll yield the floor.
Okay, Stan, those are fascinating. And I've been writing because I don't want us to not come back to all these questions. This is fascinating. Great, two really important, very different comments. Okay, so now we turn to Eric Juengst, who is a medical bioethicist. He's the director of the UNC Center for Bioethics, member of the Center for Genomics and Society at UNC, and professor in the departments of social medicine and genetics at UNC. Of note, he came to us from Case Western Reserve University, where he also directed a similar center, Center for Genetic Research Ethics and Law. And so he is now going to tell us what his biggest takeaways were from the film.
Thanks, Gail. And hello, everyone out there. Well, I had a couple of takeaways. One was I was struck by how optimistic the film was about the prospects for medical applications of CRISPR. Partly, it's because we've learned a lot in the last year since this film was made about the technical limits and risks of using CRISPR as a therapeutic tool. But the other thing that struck me was that the main worries expressed by both the experts and the great clips from Hollywood that we saw was not about the therapeutic uses of gene editing, but about their uses for enhancement. And that's really understandable given our ongoing struggle to get past old prejudices and celebrate and protect human diversity. And in fact, since this film was released, there have been a whole spate of international high-level bodies trying to figure out how to develop governance for human gene editing research. And one of the lines they've drawn consistently is to discourage enhancement applications because of the worries about what the social consequences of doing that might be. But I was struck in the film by how many of the potential human enhancements they mentioned, pain resistance, for example, or radiation tolerance, or abilities to go with just a few hours of sleep, they're likely to arrive as non-medical applications of research designed to prevent disease. So that's going to create a governance challenge for us, because most of our regulatory mechanisms are aimed at the research process, which these protocols will fare well in, presumably, because they are aimed at preventing disease after all. But then they might be used off-label, as it were, or commercialized and marketed for social purposes, for enhancement purposes. So I think that's the governance challenge that these high-level bodies and the rest of us in the scientific community will have to figure out how to grapple with in the days to come. And that's something incidentally, that we're studying now in the Center for Genomics and Society and the Center for Bioethics. Thanks.
Okay, Eric, terrific. Okay, yeah, nice plug for our research.
Right.
So next is Dr. Terry Magnuson. He is Vice Chancellor for Research at the university, and happy to have him here with us on University Research Week. It's very exciting. Terry's a geneticist. He's the K.M. and Van L. Weatherspoon Eminent Distinguished Professor, and he's the founding chair in the year 2000 of the UNC Department of Genetics. He's done basic science research in genetics, and while he was the chair of genetics, he also became the vice dean in 2010 of research in the School of Medicine. And then in 2016, he assumed the role of vice chancellor for research for the whole university. So welcome, Terry, and I'm eager to hear what your take-home messages are.
Okay, thank you, Gail. And good morning, everybody. So my takeaway from the documentary really focused on heritable human genome editing. I felt that the message from the film wasn't making judgments about whether any clinical uses of a safe and effective gene editing methodology should be used at some point. Instead, I felt the focus was on whether the safety and efficacy of genome editing technologies are or could be sufficiently developed to permit responsible clinical use of gene editing. Also stressing the need, as Eric said, for an international mechanism necessary for appropriate scientific and societal governance. The ability to make changes to the human genome with predictable effects on health really relies on a detailed understanding of how the DNA sequence variation contributes to the occurrence and risk of disease. You know, monogenic diseases are caused by mutation of one or both copies of a single gene. Examples include muscular dystrophy, beta thalassemia, cystic fibrosis, sickle cell, Tay-Sachs, and many others. Now, these diseases are generally rare, but together the thousands of monogenic diseases really impose significant morbidity and mortality on populations. Now, in contrast, most common diseases are influenced by many genetic variants, each of which have a small effect on disease risk. Scientific knowledge is not at a stage at which gene editing for polygenic diseases can be conducted effectively or safely. Also, I felt that there is insufficient knowledge to permit consideration of gene editing for other purposes, and Eric mentioned this as well. And these include non-medical traits or genetic enhancement because anticipated benefits might be offset by an unforeseen event. The final take-home point I thought was that no attempt to establish a pregnancy with a human embryo that has undergone genome editing should proceed unless it has been clearly established that it is possible to efficiently and reliably make precise genomic changes without undesired changes in human embryos. And these criteria have not been met, and further research is necessary. So one point that really stuck out to me came from Alta Charo, who was interviewed in the documentary. And she indicated that this field has moved from red, strictly prohibited, to yellow, proceeding with extreme caution, only under certain conditions. And the four conditions that I thought were stressed were, first, cases in which all of a prospective parent's children would inherit the disease-causing gene. This is the condition that I thought Alta was calling yellow, because the technology could represent the only option for having a genetically related child without disease. The second was cases in which some, but not all children, would inherit the genotype for a serious disease. The answer here is no, because pre-implantation genotyping can pick out the normal embryos. The third was polygenic diseases, not possible today. And the fourth is to enhance or introduce new traits. Also no, because as Eric indicated, really not socially acceptable at this point. So these are some of the take-home points that I had from the documentary.
Okay, wow, very interesting, and how great that all of these are different, since that was our goal, inviting you all. So the last speaker, the last panelist, is Kimberly Roboski. She is head of translational science at RENCI, at the where Stan is, and she's a bioinformatician. She also has a PhD in genetics and from Harvard University Department of Genetics. And she joined RENCI in the fall of 2016. She also has adjuncts in genetics in the School of Information and Library Science at UNC Chapel Hill. So welcome to you. Very happy to have you here and your takeaways.
Thank you so much. Quick correction, my doctorate is in bioinformatics from Boston University, but I did do my research in George Church's lab in the Department of Genetics at Harvard.
Thanks.
Oh no, thank you so much for the introduction, and I'm glad to be here. So I just wanted to sort of underscore an interesting point towards the end of the film, where the young man who was suffering from sickle cell anemia, you know, clarified that, well, if he could edit out that gene for his children, maybe he'd think about having children, he'd think about doing that. But then when he was asked, would you edit it out for yourself? He said, oh, no, 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 this is part of who I am, which I found fascinating, because I would make exactly the same choice. And I wonder how many of us would say that. And if we had one generation of everyone editing out those genes, you know, who would make those choices? How many people would say, no, I'm going to let my kids strive and struggle? You know, I'm sure that if you asked, for example, Beethoven, if you could edit out that gene that made you lose your hearing before you wrote the Ninth Symphony, would you do it? And would we have the Ninth Symphony? So these are the kinds of questions. This was my takeaway. You know, but on the other hand, in the film, there was the inevitable comparison made to Jurassic Park, genome editing in Jurassic Park, and one of the experts said that these scientists are proceeding very carefully, and the difference in Jurassic Park is that these scientists had hubris, and I would, you know, encourage people to review that movie again and tell me if you did not see evidence of hubris in some of these scientists, or even just look at their colleagues. So for sure there is that aspect of hubris, and I think it's, you know, good to enable these scientists to experiment and to innovate and to create. And I think certain Mendelian diseases, as Terry pointed out, are kind of easy targets for these sorts of things. But many diseases like Alzheimer's, you know, somebody asked about Alzheimer's. Well, there's an ApoE variant that talks about risk factor, but if you edit that out, you have no idea what the unintended side effects are. So George Church, I'm just going to wind up by saying that George Church in his lab, when I was there, he had an ethicist. So he had, you know, his 100 postdocs, but he also had an ethicist sitting in the lab, Jean-Tine Musthoff, who I had the great pleasure of sitting near and co-publishing with, to help him think about these kinds of issues. And it's for that reason that he lobbied, for example, at Congress to pass the GINA, the Genetic Information Non-Disclosure Act. So it's important to think freely and innovatively, but it's also important to invite in these regulatory experts that can sort of think about the boundaries so that the scientists don't necessarily have to think so hard about those boundaries to allow them to be creative.
I have to follow up. Do you think that that's essential during, either before, during, or after the course of these scientific discoveries?
That's a good question. George once told me, hey, look, it's not a matter of if it's going to happen, it's a matter of when. And so get ready. And if he doesn't do it, someone else is going to do it. So it's important to acknowledge that these advances are coming. So we need to get ready. We need to be asking the questions. We need to be thinking about the regulatory aspects of it now.
Great. Kimberly, thank you so much. I guess we have an agenda in which we are going to turn to some of the questions that were submitted as well. I know there's activity on the chat, but I wanted to give each of the panelists just a few minutes, if you would like, to respond to something that your colleagues said, since you've been listening to what they said. And actually, a couple of them do reflect some of the questions that we got from the poll everywhere. So I'd like to do that if I might. So Stan, is there any response you would like to make to any of the panelists?
Sure. First of all, in thinking about what I was going to comment on first, the thing that I walked away most impressed with was the bravery of the kids that were in this documentary. I mean, particularly the young man with sickle cell anemia was incredibly moving. I felt like I just wanted to hug him. He was so brave and so thoughtful, and so insightful. He was so self-aware. It was amazing to me. It was incredibly impressive. And some of the others as well seemed to me to be incredibly. They left an impression that it's indelible. And Kimberly, thank you for bringing up that comment about the fact that he wouldn't change the path of his own upbringing because it had changed him in fundamental ways. And I think all of us who are parents do feel like, the certain amount of struggle that I take on is the struggle that makes me who I am, but I'm not sure I would want to impose that same struggle on my children. And that's a deep philosophical question that I hope Eric or others can shed more light on. I also think that Terry brought up 2 points that are troubling me. One is, I think that the multiple variant issue of leading to certain types of phenotypes and disease processes is something that we really, as far as I can tell, we have not been able to attack in a systematic way. I know there's lots of people trying, I think from my perspective, at least, I view it as an extremely difficult mathematical problem, because not only do you need the data, you need to know it to a high degree of accuracy, because many small perturbations can add up to a very complex set of regulations that interact in funny ways across multiple gene structures and different proteins. And that seems to me to be a very challenging problem, and one that we would like to be able to approach. And the other thing is the side effects. I was really struck when the example was shown on the screen where they put up a cursor with a bunch of letters in the middle of it, and it was the cut and paste analogy. But when you pull up an editor for a long string, and this is a billions of letters long string, and you're going to cut a certain sequence, and you're going to cut that sequence, but you're going to cut that sequence and other stuff, as far as I can tell. I've done some reading since then, and I think that's true. Those side effects, unintended side effects, or unexplored or misunderstood side effects. And Terry, if you have corrective science to lay on me, that would be great, because to me that was one thing that I walked away thinking that I needed to know more about this.
So maybe we turn to Terry now, and then Eric. Is that okay? So Terry, you're muted.
Thank you. I can answer a little bit of what Stan was saying, but first I want to start out with Kimberly's comment about George Church. It's not if, it's when, and get ready. And I truly believe that that is true. if you look back over the last 5 years when this was really getting going in, say, human cell lines, off-target effects were happening at 50% levels, and not much work with embryos. I talked to our people that are doing this in mouse embryos, and now the off-target effects are down to about 10 to 20%, but they're not perfect. And also, when one inserts something or cuts something in the DNA, one can change the conformation and also affect potentially the epigenetics, which can affect other gene expression. So there's a long way to go to get to a point where it's used often.
Okay, Eric?
Sure.
You also can comment, of course, on any of the other comments.
Well, yeah. I had two comments, I guess. Well, a comment and a question. I appreciated Kimberly and Stan's welcoming the ethicist into the conversation. But I do that with a caveat, which academic ethicists like me are just as privileged and blinkered as anybody else in this discussion. And so it really needs to be a discussion that reaches all kinds of people and brings many different kinds of voices to the table. I'm sure you would agree with that, but it's not just a conversation for elite academics. My question is for Terry, and that was to drill a little bit into your assessment of this yellow light. I thought I picked up maybe that you were a little pessimistic about whether it's time to turn the light yellow on germline intervention or not. Or would you agree with that?
No, I actually agree with Alta in what she was saying, that it is not strictly prohibited. We are gaining lots of data using mouse embryos and other species. We're recognizing where the problems are, and it's yellow in the sense that, no, we can't do this in human embryos until we truly understand what effects that we're producing on the targeted site and also off-targeted sites. But if you look at the last 5 years, the specificity of the cuts are getting better. There are many Cas enzymes, and the ability to predict what are the best guides are getting much better using algorithms and informatics. So the field has moved quite fast.
Thanks. And I guess I would just like to ask Kimberly if you have additional comments or thoughts that you'd like to share.
Yeah, I would build off of both what Eric and Terry have said, based on what Eric said, bringing in lots of voices. I sat right next to Jantine, and I still, the ethicist at Church Lab, and I still can't tell you with great certitude that I fully understood what her job was. Even though she explained to me many times, it was about understanding, trying to ask the questions that people will ask. And so, not so much answering those questions, not so much enforcing any kind of ethic or morality on these experiments, but just asking the questions. I don't even think I understand it fully, but Eric, I would love to hear more comments on that. And then Terry, what you were saying about monogenic diseases, again, the takeaway one of the takeaways in this film was that this child had sickle cell anemia, which we know this variant is also protective of malaria. And so if you edit this out of an entire generation, and then some new strain, some new particularly resistant strain of malaria becomes, pandemic, what does that do to the human race? Well, and so this is just Yep.
No, that's a really good point. But we also know that clinically this has been done on patients in clinical trials with what looks like great success, at least over a year's period of time in this country.
Yeah,
But
and
not
I
in
think
terms-
treating as treatment is a good idea, but as you pointed out, we have to think hard about the germ cells.
Right, right.
So what I'd like to do now, if it's all right with everyone, is turn to some of the most popular questions on the poll. And it seems that the top question so far is, how can we prevent CRISPR and other emerging technologies from accelerating inequality? And I think what we would want to say is inequality in access to healthcare and ultimately inequality in health outcomes. So who would like to take a stab at that, at answering that?
I nominate Eric.
Sure, thanks. Well, yeah, it does depend on where the question is going. I'm not a real fan of the worrisome scenarios that sees the haves and the have-nots differentiating biologically because of massive use of gene editing amongst the wealthy. I think that would have to be used in such a widespread and sustained way and over such a long period that the cycles of social change will churn the haves and the have-nots many times before any population gene frequencies are affected. But the more prosaic question of whether CRISPR will face problems of access is of course real. And there, CRISPR just joins all the other innovative medical technologies. And the only way to prevent those kinds of disparities is unfortunately the hard political work of coming up with a just healthcare system and reforming the way that we do business both at home and abroad.
That's a great answer, Eric. There's a question that came in that's somewhat similar, but it also might especially speak to some of Stan's background as he was talking about the commercialization, the rapid sort of economic opportunities that CRISPR, that moving from basic science to team science that CRISPR is emblematic of. So this question is, given how cheap CRISPR is, and there's a lot, I can't remember if the movie really emphasized this, but it's quite a lot cheaper than other, gene modification modalities, for sure. And given how cheap it is, will we be seeing sort of proliferation of garage labs where people do it yourself, or adoption by commercial direct to consumer marketing companies that may or may not be contribute to inequality? So Stan, can I entice you with that question?
I'll give it a shot, but I think that perhaps others will have even more to add. Well, I'm sure they'll have more to add and more specificity. So I do think that the technology is reasonably inexpensive to demonstrate, but to be very precise in applying the technology, I think it requires a fair amount of infrastructure and understanding, laboratory infrastructure in order to implement, particularly at scale, where you're trying to propagate the effects in a significant way. I absolutely, there's companies here in North Carolina that are using CRISPR and the techniques to look at problems that are of benefit to society, like better crops and different types of food processing mechanisms. So there's the amount of possible applications are just profound. And some of those were in the documentary. I mean, North Carolina showed up. And it's a point of pride, I think, for us that we in the Triangle, as well as across the state, have made early investments in the biotech industry, and we stand to reap some of those benefits as this technology develops. And that's one of the reasons that we see a certain uplift in our economy that I hope we can sustain by graduating more smart young people that have an ethical understanding of the fundamental problems. I do fear, honestly, that there are places where in the garage things could happen that either go awry or have been designed to be malevolent. That gives me pause. It was mentioned in passing, but our colleague here at UNC, Fernando Pardo Manuel, has schooled me on a couple of occasions about the idea of gene drives, where you, and Terry can tell us, and Kimberly can tell us about these. I was just stunned when I heard about this concept of using CRISPR to basically preclude a certain evolutionary pathway in the future. Those kind of things give me great pause and concern. I know I've gone all over the place with the answer, but I think that we're going to see industries grow up around this. They're going to be in all fields, better plants, roses of new colors, wheats of different protein. As a sourdough baker, I'm always looking for a new flour. I'm not a very good sourdough baker, I might add. But there's from the prosaic, but also to the profoundly important in treating diseases where, again, this inequity, our inability to provide it to everybody on an equitable basis bothers me. There's a lot here. There's just a lot here. This is a technology that we have got to wrap our heads around because it's here. So I'm sorry to talk so long.
No, that's great. You've raised a number of different things. Would others on the panel like to respond to?
Sure. I mean, some practical, when we talk about North Carolina, practical applications, a company Engagetigen, I don't know if I'm pronouncing it right, using CRISPR to make allergen-free peanuts. Locus Biosciences using CRISPR to engineer viruses that will kill bacteria in people because we're losing antibiotics. Epigenos, who's using CRISPR technology in cancer to suppress genes that should not be expressed. Syngenta, I thought, was mentioned in the film as well. So there are a lot of practical uses that will bring good things.
And these are not germline, or are they germline modifications?
Yes, I think what we're talking about, like allergen-free peanut, that would be.
Okay.
So but in plants, not in humans.
Yeah, in plants.
Right. So Eric and Kimberly, any thoughts from either of you about this topic?
If I may, yeah, I mean, synthetic biology has been around a long time. And when I was in Church Lab, we were working with zinc fingers and talons for doing this kind of genetic editing experiments, but they were super expensive and they took months to generate. And so when that MIT paper hit, it made huge, it was like a depth charge going off in the lab, and people were very, very excited because it was so cheap and so easy to do these kinds of synthetic biology experiments that have been around for years. I mean, you can go and buy a neon tetra from Glowfish that's, been edited to look pretty in your fish tank, but you're advised not to flush it because it might go into the food chain. So these are the kinds of things that, it's already here. And now it's going faster, and we just need to think hard about the questions like, okay, but what about human germline, and somatic treatments and those things.
Okay, and Eric? No, muted. You're muted. Yeah.
No, I think I'm good. I mean, just reiterate the point that we have, the scientific community has a pretty good grip on the governance of research. Except that when it's garage research. And so the whole phenomenon of citizen science and DIY, biosynthetic biology, and it is very interesting phenomena to watch and wonder about how do the controls apply in those circumstances.
Right, right. We have, not in this film, but it can certainly be found on YouTube. Josiah Zahner, the most famous probably do-it-yourself garage guy, who does have a PhD in, I think, biology. He injected himself. He made a potion and injected himself on camera. So that's out there in the world. Okay, so I'm not asking you all to comment on that, but I do have a question that I think incorporates a couple of the chat questions and also brings us back to the negative outcomes and Terry's comment that in mouse embryos that the off-target effects, that people were certainly improving those outcomes, so they're not eliminating them. So here's the question, and again, please, all of you take a crack at this. The film did a great job of linking the science of CRISPR to some of our greatest fears, which are expressed in literature and film about science running amok. So what will protect us from negative outcomes from these? Are we to think, well, we're just gonna accept that we can't control off-target effects entirely? And you guys have raised the question about using gene drives for modifying animal populations or crop populations. So who's going to oversee this? Who can help with this need to go red or go yellow, as Altacharo said? And do you trust those oversight? Where are those bodies? Is it WHO? Is it national? Is it local? Is it our IRB? Do you trust them to protect? So it's a series of questions, but you get the general gist of it, which is science run amok versus no, we really can work on this ethically and in a way that it will be protective.
So, Gail, if we look historically, go back to 2004 with human embryonic stem cells and the worry about cloning humans and so forth. So our federal government refused to do anything about it and wouldn't allow new stem cell lines to be made, but it wasn't really regulated. And so what stepped in were the national academies that spent, literally 6 years writing the guidelines for use of human embryonic stem cells relative to treatment, to cloning, etc., that were adopted by the entire world. Universities and research centers, they certainly weren't required, but people or universities adopted them. In the film, I do believe they're talking about an international commission, which would be along the same lines, providing guidelines, but no enforcement.
Right. I think, as all the scientists out there know, science has a lot of oversight when you're working, especially with people, from institutional level IRBs and biosafety committees on up through NIH and the FDA. So, and we can combine those kinds of hard regulations with the soft regulation of the scientific community itself, its own self-policing and thinking through the national academies. That seems to be a plausible picture for providing protection. What I'm interested in and worried about, I guess, is the fact that this is no longer siloed within countries. Science is global, and so the solutions have to be global. And how in the world will that work? Whether a global scientific community can have enough clout to do what the national academies have been able to do in setting professional standards, or whether it will need some sort of harder governance. that's still very much open.
Kimberly,
I agree, Eric. That is the question. Global regulation would almost be impossible.
I have a comment to make, but I want to defer to Kimberly if she has, because she has a greater degree of insight. And I think you and I have talked in the past, Kimberly, about some of the concerns that George Church put forward too. You're muted.
Yeah, you know, so there was a very active do-it-yourself bio component in Church's lab, you know, where George invited brilliant people to just come and be brilliant, and, you know, sort of step back and gave them the tools to invent all kinds of wonderful things, and operationalize things like the CRISPR-Cas9 system. And I also sat next to one of the DIY bio people, and I also was part of a citizen science delegation to a UN effort last year. And so I kind of am hooked into that, you know, garage science mind share. And I do honestly have to say I have concerns about that. But also, you know, it is kind of democratizing in a sense that you can allow some brilliant high school student to come up with better understanding of genomics and contribute to the our genomic understanding, as well as someone who has access to the IP in, you know, in some large corporation. So in that sense, I'm very optimistic about it, you know. And then the other side of the coin is that, you know, I'd like to ask Terry what he thinks about this, but I think in a way this is sort of a self-regulating operation, because, you know, Mother Nature is going to get in the way. I mean, there are only so many ways you can tweak the genome, and the thing that you tweaked it on is going to survive. And so, you know, yeah, there's, you know, all kinds of scary things that can happen, but life is scary and risky. And, you know, I mean, so many years of evolution is going to put some guardrails on it for us as well. What do you think about that, Terry?
Yeah, I agree. In the mouse world, we don't really worry so much about off-target effects because we can just breed them away and keep the one that we want. In the human world, that's a little more difficult, but can happen. So those would be guardrails.
Yeah, and
I
in
think
terms of the gene drives, sorry, go ahead, please.
Yeah, I think I worry more about what's happening at the target site.
What do you
Can
mean
you
by
elaborate
that,
on
Terry?
that? Yeah.
Pardon?
Please
Well,
elaborate.
in terms of putting in new sequence, making a deletion, whatever. Again, I mentioned you can be impacting the three-dimensional structure of the chromatin, and all of a sudden you could find that gene next to a different kind of enhancer, and it's being expressed in ways that you never thought about, which could lead to gene drive. I don't think we have enough information on that.
Yeah,
So,
I would. Go ahead,
so
Stan.
Terry, is it true, or maybe I should ask this question to everyone because I don't know. So is it true that by making this technology available, we are speeding up the mutational probability along a lot of different dimensions that perhaps would not have occurred, or would not have occurred in as rapid a fashion in, you know, a world without without CRISPR and Cas9? So I mean, can we look at it as you can argue that it's a consequence of evolution because we got here by a process of evolution, but is it true that we're going to accelerate the possible you know, variants that get propagated throughout the ecosystem? Is that an irrational thought?
No, it is rational. In mouse, genetic drift actually happens quickly, fairly fast.
Terry, would you define
But-
genetic drift for maybe some
Genetic drift, you know, for example, if you get an inbred strain from the Jackson lab, C57 Black 6, you bring it in your lab, you breed it for a year, it turns out it will be very different than the Black 6 that's at the Jackson lab because of spontaneous mutations and alterations that occur.
in all humans and animals?
Right.
Okay.
But surely these man-made changes will be a drop in the bucket compared to the other kinds of forces that are shaping our evolution.
Yeah.
War and starvation and climate change.
Environment and how environment is impacting epigenetics, impacting gene
Right.
expression, all kinds of, leading to diseases. And
yeah,
I mean, that's one of the big major focuses now is the exposome from birth to death, both externally and internally.
I have to ask you to define that.
Well, that exposome is what you're being exposed to, you know, in the environment around you, but also inside in terms of what kind of diet, what kind of medication, what kind of water you're drinking.
Okay, thanks.
Whether or not
We
you live
call
in
it the
a
environment.
neighborhood. Yeah, and you're in a neighborhood that does not have access to good food and fresh food, or you're in a neighborhood of, I mean, it's pretty easy to show that you're living in a neighborhood that has a high number of gunshots per year, that your mental state in any given time is different than people who live in a quiet neighborhood. So the inequalities of all of these questions are, that to me is something we need to spend a huge amount of time thinking through, because as a society, we face a lot of challenges.
Well, can CRISPR be used specifically to address that? Can there be genetic engineering that happens to address, or are we talking about social change, social determinants that CRISPR's not? CRISPR's way down the pipeline of.
I think so.
Okay.
I suspect so.
DNA variants alone, I mean, that's important, but that's not the answer.
Okay. Now, we do have a question about whether genetic engineering or re-engineering the population might occur with CRISPR, and how do we prevent it from becoming a modern eugenic movement? And to a certain extent, you all have talked about this, but here it is. How can we ensure equal access to the technology and not promote ableism? So that would be, you know, promoting the most fit. Well, I think probably most people know what eugenics is, but selecting for the most fit, the most whatever, the best. Eric, I know you've thought about this a lot. Do you have anything else you'd like to say about that?
Yeah, I think we've touched on it a number of times, the difficulty of actually changing population gene frequencies with kind of sporadic interventions we'd be making. It really would require a massive eugenic effort to do that. And the problems of assuring equal access to technologies. I guess the only new part I would add is that at least in democratic countries, I don't think we're likely to see a state-sponsored eugenics revival. Cross our fingers. But the effect of the commercialization of prenatal testing and genetic interventions could produce a kind of consumer eugenics, where fads and fashions drive a lot of people's decision making in a powerful way. So that's something to look out for.
Okay. Now I realize it's
To follow up on that one,
oh, good. I was going to ask if people want
is one might be able to do that for eye color or whatever, but as indicated in the documentary, you want to make someone smarter. Well, you don't even know
Right.
where to go to do that.
Right.
Yeah. And we are almost out of time. And so, Kimberly, have you got some thoughts that you'd like to leave us on this?
I would just add what I heard from Nobel Prize laureate Sir Richard Roberts when I was at Boston University. I had the great privilege of having wonderful mentors. And he said, your grandchildren aren't even going to understand the human genome. Go study E. coli. And so, you know, to what Terry said, we don't even understand how eye color works yet. So we have a long way to go.
Mm-hmm.
So maybe that's our biggest protection. You think?
Yeah. I think so.
And so could I ask a final question, just as with the prerogative of a moderator, that, you know, this is title of this, CRISPR, the most important scientific discovery of the 21st century. You know, maybe something about E. coli ought to be in there. But my question about CRISPR is, it seems, is it a serendipitous kind of major breakthrough and having all these special kinds of characteristics because it came from nature, it came from somebody observing bacteria. And then it, as Stan said in the very beginning, it just sort of moved along the rapid pipeline from basic science to team science, translation, et cetera. Is this unique? Is that why we're talking about CRISPR now? Or is this what happens with science? All of you know science. And what would you say about that? Is this just another interesting science documentary?
No, I think it's been a long road. I mean, you go back to when I was a graduate student, we're trying to figure out how to do homologous recombination to change genes. Mm-hmm. And, you know, that was a long road to get to a point where we could do that. And then the talons and so on and so forth. It's been a long road.
Okay, thank you, Terry.
But the serendipity is part of normal science.
Mm-hmm.
So that's not an unusual feature, I think.
Sorry, what's an unusual feature?
It's not the serendipity involved in, you know, finding this in the salt marshes in Spain. That's not an unusual feature of normal science.
I totally agree with Eric, but this one was a sweat equity. And this breakthrough is like the invention of the transistor. In my opinion, this is really a big deal.
Wow. All right, Stan, you could have the last word on this.
No, I completely agree with Kimberly. I think you can see evidence in the past of scientific, you reach a certain level of knowledge about how things work, and then suddenly somebody figures out that one little piece like the transistor and and then suddenly you have, you know, what we think of as the modern era with computers and everybody carrying a massive computer around in their pocket. That's a big change. It's changed things from, you know, across the spectrum of society. And I think this is an equivalent of that.
Okay. Well, last word is very eloquent. Thank you, Stan. Thank you, everybody, for your participation. I think I learned a lot, and I'm supposed to tell you now that there will be a recording of this. It's available at the same link that the place where you registered this. And great celebration of Research Week at our wonderful university, where a lot of this science is really taking place, and ethics. Okay, so thank you again, and I think this is it. It is exactly 12 o'clock. How's that? Perfect.
Thank you, Gail.
Thank you, Gail.
Okay, well, thank you all. This was really fun. All right, bye-bye.
Bye-bye.